be presenting to you today about low temperature solutions for oily soil removal in laundry. So just to give you an overview of what I'll present, we'll begin with our challenge and give you some background on why this challenge is important to our clients. And then we'll go over 5 new strategies that we've come up with through our project to address this challenge before bringing our presentation to a close. So our central challenge is to identify safer, sustainable, and effective solutions for oily soil removal in laundry at low temperatures. We'd like to take this moment to thank our partners. These are the partners who brought us this challenge, beginning with Method and 7th Generation, both of which are consumer product companies specializing in cleaning products and cutting-edge environmentally preferred products, and also BioAmber and Amyris, 2 innovative chemical companies who use renewable feedstocks to create greener chemicals.
So, as Akash mentioned, our partners presented us with this challenge. Before we talk about our strategies in more detail, I'll give you a little bit of background about why oily soil is a problem and why it's ideal to remove it at low temperatures. There are 3 types of oily soils: body soils, food and beverage soils, and organic materials such as clay and soil particles. And as you can imagine, these different compounds can agglomerate together and create very complex soils that are difficult to remove from clothing. 3 types of energy are at our disposal when we remove these soils in laundry: chemical, thermal, and mechanical. And it's easiest to remove oily soils from laundry in hot temperature washing because it helps make the oily soils slightly more soluble than they would be in cold water. It loosens them from the textiles. But if we lower the washing temperature, this greatly reduces energy consumption, and it's one of the biggest impacts that we can have in the laundry process. Globally, we're seeing trends in Latin America, Asia, and Europe where consumers and manufacturers are moving towards the direction of using less and less water, as well as using colder and colder water for this impact to reduce energy. So our design criteria in this project was to investigate how we could enhance current Method and 7th Generation laundry detergents to be more effective in cold water washing. Cold water wash, which is simply unheated water, depending on where you live, that could be between the range of 5 and 20 degrees Celsius. We also wanted to prevent redeposition of suspended soils, to use ingredients that degrade readily in the environment, that pose a low toxicity to humans and the environment, and preferably to be sourced from renewable feedstocks. And we were constrained by the fact that we're simply looking at liquid laundry detergent. We are not changing the current washing machine technology. In other words, we're not changing the mechanical energy input that goes into the laundry process. We were cognizant of cost, and we were also avoiding problematic ingredients such as EDTA, phosphates, VOCs, and others that are harmful to the environment or to human health. So, how does laundry detergent work? I'll just briefly introduce some of the main ingredients that we'll be talking about later throughout the presentation. So, surfactants are very important in laundry detergents, and as you can see here, they have a hydrophilic head and a hydrophobic tail, and they can assemble themselves around oily soil on the textile, and then when they completely go around the textile, that's called a micelle, which then facilitates the removal, the lifting up of the soil from the textile. Solvents help break up soils and make them more soluble in water, especially oily soils that would not normally be soluble in water. And then dispersants help those oily soils stay suspended in the wash water and prevents them from redepositing onto the textiles so that they're washed away. And then lastly, enzymes chemically degrade soils, making them easier to remove. And here are some examples of where enzymes can break bonds to break up sugars or proteins or lipids into more elementary building blocks to make them simpler to wash away. So next, Kira will be discussing our hazard assessment.
So we examined the ingredients that are in Method's current Laundry 4X detergent. And this is representative of many other environmentally friendly consumer laundry products. So of the surfactants that are used in the current formulation, we found some concerns with skin and eye irritation as well as some low to moderate aquatic toxicity. However, for the solvents that we looked at, we found relatively low risk and low concern. But we just wanted to point out that limonene actually, that Method actually uses as a fragrance in their products, but has some interest and application as a solvent. We found some aquatic toxicity, respiratory irritation, as well as other issues around indoor air quality. So although we're not addressing fragrances in the product today, or in the presentation today, we just wanted to point that out. And it's not something that we would recommend as a solvent for use in these products. So going back to our challenge, we approached our challenge on 3 different levels, which can be thought of as kind of a hierarchy of scale. So the first one is a chemical level approach, which means we consider some direct replacement of some of those current ingredients in Method's current product. We then also looked at a formulation level approach, which means we thought about how different components and chemicals within the laundry detergent might interact to more effectively remove oily soils at low temperatures, as well as, again, be sustainable and safe. And finally, we took a process level approach, which was kind of us stepping back from thinking about just laundry detergent in a washing machine to think about what other ways could we clean laundry at low temperatures. So with that, we came up with several strategies that we'll present to you today, and we'll explain how they each fall along this spectrum of approaches in some way, and we'll describe how they address our challenge at each of these technical levels. It's a little light,
Yeah,
but
can we just take a timeout moment and adjust the brightness on this, because we're missing some of the detail that I'm meant to see.
So while we're doing that, the limonene is actually, it's just a small component of the fragrance. It's not actually a solvent. So
Right.
when, for labeling purposes, you list those things that are on certain lists.
Right.
That's on
Great. So our first collection of strategies were inspired by natural deputectic solvents, or NADES. And NADES are bio-inspired, and that they're hypothesized to act as an alternative media to water in organisms. Specifically, they're made up of chemical components that are abundantly found in nature, such as sugars, amino acids, or organic acids. And when these are combined in certain molar ratios, they exist as a liquid with unique properties. And it's believed that these NADES in nature are able to solubilize, store, and transport materials that are poorly soluble in water, such as lipids. So the word eutectic in the name, natural deputectic solvents, means depressed melting point. And this occurs due to hydrogen bonding formation between the chemical components of the mixture. And this results in a chemical mixture that has a melting point that is lower than any of its individual components. NADES in general have a very wide range of depressed melting points based on, again, their chemical makeup and their specific molar ratios. But this is just an example of a few that do have depressed melting points that are right around the temperature that we're interested in for low temperature laundry. So this was definitely something that inspired us to take a look into these solvents a little deeper. So because of this hydrogen bonding, this allows for NADES to incorporate water molecules into their structure as well. And it's been shown that the incorporation of water affects the properties of NADES in several ways, such as changing its viscosity, conductivity, and of course the polarity of the solvent itself. But one question that remains unclear at this point from the literature is whether or not NADES maintain their structure as well as their unique properties in high quantities of water. or whether or not they might dissociate based on the water molecules kind of competing for the hydrogen bonding between the two chemical components. And in that case, the chemical components might just exist separately in an aqueous solution. This is definitely of interest to us for if we were to use NADES in a laundry detergent, where in a washing machine there would be a huge influx of a large quantity of water. But exactly how water affects NADES is still unclear, and more experimental research needs to be done with specific NADES compounds to better understand this. But another thing of interest for us is that NADES are able to solubilize hydrophobic materials. The graph on the right is representative of a NADES that's made up of 1,2-propane-diol and choline chloride. And it shows that at 0% water content in its pure form, this solvent has a high level of solubility to solubilize two very lipophilic compounds. But again, as water content might affect these solvents' properties, as water content increases along the x-axis here, for each of these compounds, you can see that the solvent becomes less effective at solubilizing these really non-polar hydrophobic compounds. The exact water content at which this kind of drops off is definitely different depending on the solute of interest as well as most likely the NADES itself. So something to keep in mind in terms of how water will affect these NADES function.
So we propose using NADES in three potential ways. So the first way is to use them as solvents in and of themselves, in the same way that solvents are already used in laundry formulations to help break up and disperse greasy stains. So on the left-hand side, there are 4 compounds that are already known from the literature to form NADES, and on the right-hand side are 4 compounds that have never been tested as far as we know that we propose should be investigated. Now, as to why we chose these particular compounds, there are 2 main factors. One is that a lot of the molecules that have been tested in the literature as NADES are sugars or things that are nutrients, and we wouldn't necessarily want to add those to a laundry formulation so as to prevent unneeded microbial growth and things of that nature. Another really common component used in the literature to study this phenomenon is choline chloride, which Kira mentioned. But we wouldn't really want to use choline chloride because it's the only compound in this set that is a quaternary ammonium salt, and that might have undesirable interactions with other ingredients in the laundry formulation and might also have some unwanted bioactivity. So we have picked a number of chemicals from the literature that are already known, and these are all quite safe. In fact, glycerol is already used in laundry formulations as a solvent by itself. Citric acid is commonly used as a preservative, malic acid is used in food, and proline occurs naturally as a constituent of protein. As to new NADES components that we're proposing, perhaps one of the more interesting is succinic acid, because as to sourcing this ingredient, one of our partners, BioAmber, happens to manufacture succinic acid from renewable feedstocks. So this would be an ideal opportunity to combine some of the interests of our partners. Other molecules that we propose testing, we arrived at these just from the theory in the literature that NADES are comprised of a hydrogen bond donor and a hydrogen bond acceptor. And although it's not entirely clear sometimes how molecules behave, we anticipate that protonated amines like these would act as hydrogen bond donors, so that might be a useful component in the formulation, whereas these might act as hydrogen bond acceptors. So that was one strategy, simply adding them to the formulation as a solvent. Another strategy is modifying the structure of NADES themselves and maybe trying to tune their properties so that instead of having hydrogen-bonded collections of molecules that act as solvents, we might have a hydrophobic part amongst that hydrogen-bonded assembly, and that would perhaps make them act as more like surfactants or dispersants. So we propose experimenting, and this, I stress that this has never been tested before, experimenting with adding C8 to C10 alkyl groups to some components of nades and seeing if they form nades that have this alkyl group in them. So that could easily be accomplished by making esters with these acids, malic acid, which is known already to form nades, and succinic acid, which, as I mentioned, is conveniently available from bioamber. And then these resulting alkyl esters can be combined with other components to form the new putatively surfactant-like nades. So this is something definitely to test. It's quite experimental. A third strategy, since, as you know from what Kira said, the properties of nades as solvents depend on how much water and how dilute they are in water. It's clear that the less dilute, the more concentrated they are, the better solvents they are for hydrophobic components like greasy stains. So we propose to make a pre-treatment product based on nades that would be highly concentrated and would immediately go to work once applied to a stain, taking out that oily soil in advance of washing in laundry. And this could potentially greatly improve the stain removal in subsequent cold washing. So those are the three strategies. And Kira will talk about the hazard properties of nades.
Great. So in terms of human health concerns for nades, we would expect them to have low risk because a lot of these components are products of cellular metabolism and occur naturally in humans. But there have been a few studies that have looked at nades and their toxicity experimentally, and these have usually been on choline or mostly choline chloride based nades, but they are somewhat informative. So several studies have found that they have low to moderate cytotoxicity as well as low phytotoxicity. And definitely depending on the nades, it's obvious that the toxicity is likely dependent upon its chemical makeup and what exactly those individual chemical components are. But one interesting finding from one study was that the toxicity of the nades mixture was lower than the toxicity of its individual parts in solution. So one thing to keep in mind. But biodegradability is also an important consideration in thinking about the environmental impact of this chemical or this chemical mixture once it goes down the drain. One study found that 3 different choline chloride based nades, one with glycerol, one with glucose, and one with oxalic acid, were all readily biodegradable, which means that more than 60% of the compound was degraded within a 28-day period. But as you can see from this graph, which is from another study, the biodegradability of nades might vary widely, again, based on its chemical makeup. So another thing that would have to be tested experimentally depending on the specific nades. So we think that based on the potential, or the evidence for the potential of these solvents to have relatively low hazardous traits, we think that the 3 strategies that Akos described might be interesting and promising avenues for our partners to explore further.
Thanks, Kira. Moving on to another strategy altogether. I mentioned that our partner BioAmber manufactures succinic acid, and they also manufacture alkyl esters, dialkyl esters of succinic acid. These organic compounds are made from renewable resources and have solvent-like properties, so these could be potentially used as solvents, simply as drop-in additions to laundry formulations. So we decided to explore how that might work, and although there's a variety of different dialkyl esters that can be made from succinic acid, a really great variety, we've just focused in on 5 compounds which roughly fall into 3 categories: short-chain esters, medium-chain esters, and long-chain esters. We think that these are representative examples of those 3 categories. Solvents interact with soils in a variety of ways, and it's actually quite complex and interesting to think about what makes something a good solvent for a particular soil. There's actually a way to find that out numerically, and it's called Hansen solubility parameters. It's widely used in formulation-based product industries. Hansen solubility parameters break down the intermolecular forces that are exhibited by a solvent into three categories: polar, dispersion, and hydrogen bonding, which are three different patterns of chemical interaction. And by numerically separating these, you can describe how a solvent acts on its other molecules that it's surrounded by. So we looked up, there were literature values available for the Hansen solubility parameters of dimethyl succinate, and for the other four compounds, we calculated them based on an estimation method that basically breaks down the molecular structure into chunks, and you can add up what each chunk represents as a contribution. So without delving into the actual numbers themselves, the thing that I'd like to point out here is that the HSP values for these solvents are quite similar, or at least in the same ballpark, as a number of soils that are highly relevant for laundry. Carbonized residue, which corresponds to things like burnt food, cottonseed oil, which is made of fatty acids, olive oil, also fatty acids and triglycerides, and saturated fat, or lard. So the fact that there are some similarity in the HSP values for the soils and for these compounds suggests that they might actually work as solvents. That would really need to be tested empirically, though, and we have not done that, so that is the next step that we propose for our partners. As to the hazard properties of these solvents, which we also evaluated using literature searching and computational tools, there is not a huge amount of literature available on the human health effects of these compounds. There's some limited evidence of acute respiratory toxicity for dimethyl succinate. But besides that, the fact is that these solvents are not volatile organic compounds, VOCs. They're more like semi-volatile. So in normal use, one would expect very minimal consumer and occupational exposure to the solvents from inhalation. So that makes it a bit less of an issue that we have limited evidence of this toxicity. They also have quite low flammability as measured by GHS criteria. Their environmental fate, we expect them to be readily biodegradable, but the only evidence we have for this is modeling and prediction. If they are not biodegraded, then they could be persistent. We don't really know that. We only have an estimate of the overall persistence from an environmental model. But we also expect that they're not bioaccumulative. They don't generally have the kinds of properties that confer bioaccumulativity. The long-chain esters might, but at the same time, we expect them to be metabolized readily so that they wouldn't really fulfill their bioaccumulative potential. In terms of our overall map of strategies, this would basically represent some kind of a drop-in chemical substitute or a simple addition to the formula. Nothing really involving a rethink of the product design or the formulation. And I'll hand it over to Julia for the next strategy.
So our next strategy enlists biosurfactants, or surfactants that are of natural or biological origin. And there's a wide variety of bacteria and fungi in nature that secrete surfactants for a wide range of purposes, including solubilizing hydrocarbons and oils so they can digest the material and for nutrient intake. They can attach and detach from substrates, hydrocarbon substrates, to facilitate growth in a variety of different environments. And they can even organize community-level biofilms, which are these large microbial communities with pores or channels that allow for hydration throughout the community and nutrient dispersion throughout the community, waste disposal, and those on a larger scale aid in things like petroleum spill remediations. Glycolipids are one class of biosurfactants that include rhamnolipids and sapphorolipids. Both rhamnolipids and sapphorolipids are formed from carbohydrates and hydrocarbon tails. The rhamnolipids are generally synthesized by bacteria, and the sapphorolipids are generally synthesized from yeast. Another difference is the sugar molecule that's attached. So for rhamnolipids, it's a deoxy sugar molecule, and for sapphorolipids, glucose disaccharide. And so the carbohydrate here is the hydrophilic head, and the hydrocarbon is the hydrophobic tail that we talked about earlier for surfactants. I want to point out this structure, the lactonic sephorilipid structure is a closed ring structure. You can see here it's closed, versus the acidic sephorilipid, which is not joined on the right there. And there are some studies that show that some biosurfactants, like the lactonic sephorilipid, can form 3D structures. So we talked about micelles earlier. They can also form these cylindrical type shapes, other 3D shapes, including these multi-layered micelles or lamellar vesicles that are actually used currently in liquid laundry formulations to solubilize insoluble ingredients. So this might be these biosurfactants that can form these 3D structures might be particularly useful going forward. Another kind of surfactant found in nature is cyclic lipopeptide called surfactant, which is a natural antibiotic from a specific bacterial strain that has been found to be very powerful in terms of its ability to reduce surface tension. It's also been shown by one study that I thought was really interesting to have really good wash performance additively with other ingredients in detergent at low temperatures. So I thought it could be very useful for our particular energy saving challenge here. And it also sort of begs the question of whether we can also seek formulation approaches for biosurfactants like we are proposing to do with the deep-ejectic solvents. Biosurfactants have gained a lot of popularity over the last 10 to 15 years over non-renewable surfactants because of their high-performing ability, but also because they've been shown to be more degradable and less toxic than synthetic surfactants. And you can just see with this graph over the last 10 to 15 years, the number of publications this shows for sephorilipid has increased dramatically. However, the biggest challenge to date, and it still remains a challenge, is large-scale production in terms of cost and scaling up. That said, there's a lot of research going into currently and recently into how to make this process more cost-effective. Companies like eCover and academic researchers like Jeannie Pemberton at the University of Arizona are working quite a bit on how to optimize production for the future, and some companies are actually using surfactants and or rhamnolipids specifically in their products currently. And this is a yeast model from one of our chemical partner companies, Amyris, that they use to basically break down sugar biomass into elemental building blocks that they then reassemble with this genetically altered yeast metabolic pathway to create a variety of different compounds. And we thought this could be a really promising way for them to produce their own version of biosurfactant in the future. And we thought that sephorilipid might be more promising in this realm because of they're naturally synthesized by yeast. But this would take more research to figure out whether this is the right model for this purpose. And the idea here would be direct level ingredient substitution from the existing surfactants in the method 4 times laundry detergent with rhamnolipid or sephorilipid. And we think that they score pretty well on toxicity. I just wanted to go over this briefly. As I said, they're readily biodegradable, as has been shown by standard guidelines. Low cytotoxicity in human cells has been shown. Lower skin and eye irritation compared to conventional surfactants, and they have been identified as exhibiting aquatic toxicity, which is a lot of the chemicals we've come across is kind of the main concern, I would say. But they've been shown, at least in 1 or 2 studies, to be a little bit lower on the aquatic toxicity scale than some conventional surfactants. But there are existing data gaps. Even with these data gaps, we think that biosurfactants could be a really promising avenue for direct level substitution going forward, but also maybe possibly even this formulation level approach as we saw with surfactant.
Okay, our last biologically inspired design we'll discuss are enzymes. In nature, enzymes degrade proteins, sugars, and lipids by breaking bonds. They're produced in our body. They're also produced by bacteria, fungi, and many plants. Bacteria are used to commercially produce enzymes such as protease, amylase, and mannanase, which are currently used in method and 7th generation cleaning products. And they're used in combination because each class of enzyme targets a different type of molecule, and they can break apart these complex stains that are difficult to remove. Lipase is another type of enzyme that breaks down lipids, and they hydrolyze the triglycerides seen here into glycol or glycerides and free fatty acids. So they break apart the three hydrocarbon chains that are stubborn to remove, such as kitchen grease that might get splattered on your shirt. And we feel that lipase may break down fats more effectively at low temperatures and also in alkaline environments than current ingredients. Although, lipase is not currently used in our partners' cleaning products, but there are a number of laundry applications where it is being used. Some of the technical constraints of using lipase is that it has a slower reaction time than amylase and protease. A malodor or an unpleasant smell is produced when butyric acid is produced as a byproduct in the breakdown of triglycerides. It has a long duration of activity, so it continues to work into the drying cycle. This could be effective in terms of removing all of the lipids and breaking them down, but it also continues to produce a smell, the unpleasant smell, which we don't want in our laundry. So one of the reasons that it may continue to be effective in the drying cycle is because it works at an optimum water content of 20 to 30%. Given those constraints, we feel that there are opportunities to use lipase, given some of the recently engineered strains of lipase. Lipex is produced by Novozymes to be most effective in the first wash, and it's also highly effective at 20 degrees Celsius. And a paper that was published in 2013 evaluated this bacteria strain, Staphylococcus aureate, that produces lipase that is synergistically effective with non-ionic surfactants and with oxidizing agents such as bleach. So you can see in the graph on the right that the top line represents the interaction between non-ionic surfactants and lipase that produce a much higher oil removal percentage than the individual ingredients on their own. Process and formulation changes can also present opportunities for using lipase. So we feel that using lipase in a pre-treatment solution might be very effective because it would have the optimum water content of 20 to 30%, and it would also have a longer contact time on the textiles, so the slower reaction time would not be as much of a problem. We could turn that potentially into an advantage. And synergism and antagonism can be used also in formulation to use ingredients with lipase that could either inhibit its activity to slow down potentially or stop its activity, or to enhance its activity. For example, protease and lipase interact synergistically as well. Overall, enzymes pose a low risk of human and environmental toxicity. Lipase specifically is even safer than protease, although enzymes do show evidence of respiratory and dermal irritation and sensitization. However, some of this data comes from occupational exposures that were very high-level exposures from the 1970s, and currently formulations have been changed and consumer exposure is very low, and it is also estimated that environmental toxicity is very low, and there's no evidence of biodegradation. So this is where enzymes would fall on our spectrum. We feel that there are opportunities to add it into a formulation in conjunction with other ingredients, and we also feel that it could be used as a pre-treatment and the process level of how we clean our clothes. Potentially it could be used with NAIDs or with biosurfactants to be highly effective at removing these oily soils.
Thanks, Molly. So the last strategy that we'll present today is completely outside the box, and within the confines of our scope of work is our attempt to try to rethink laundry a little bit. Forgive me for stepping back a little bit. The inspiration here is, the bio-inspiration here is surfaces in nature that are capable of repelling water and dirt very effectively. This is accomplished, for example, in leaves, not by the chemical nature of the coating on the leaf, but by the shape, the roughness of the surface itself. So you can have leaves that have super hydrophobic surfaces, and chemists recently have also been able to mimic that roughness chemically and create things like this super hydrophobic paint. Also to create super oleophobic surfaces, which repel oil instead of water. So this is a rich area of research, and just one tiny cross-section of this research we found to be potentially relevant to a very strange application. So it's possible to create a multi-layered polymer that has functionalized surface so that you can add this R group, being whatever you want, onto the surface of the polymer, and that will then be the chemical that will confer some chemical character to the surface in addition to its shape. So you can have that shape, roughness, and different chemical characters. And this has been used to create surfaces that are super oleophobic and also oil adhesive at the same time, so that oil does not coat them as it usually does things, but it just sort of sticks to them in droplets, and it even works underwater. So here's an example. You have these 3 droplets of a greasy substance, and that one droplet in the middle is on the only part of the piece of glass that's not coated in an oil adhesive surface. And then when you tilt that piece of glass, that drop rolls off, but the other 2 stick, and they don't move. And this is happening underwater. So an interesting application of this technology is to create a filter where you can separate oil and water by gravity filtration through materials like this. So we thought, what could you possibly use this for in laundry? Well, one thing you could use it for is to collect oil that has been removed from clothes and is dispersed in solution and potentially in danger of depositing back onto clothes. And that could easily happen at low temperatures. So we're proposing a totally new product that would be added to laundry, and it would be some kind of durable, reusable object that is liquid permeable and has a really high surface area. So something like this, it could even be 3D printed, for example. And it would have a super oleophobic oil adhesive coating on the inside surfaces, not the outside, so that you don't want oil droplets touching things on the outside. So this would collect oil from the laundry wash, and then at the end you take it out. It's just a physical object. Eventually it would lose effectiveness and need to be regenerated somehow. So this is the concept. So thinking about what this concept means in terms of sustainability and environmental health, a number of problems show up. One is that we're creating a new durable object, so that requires some kind of product life cycle stewardship, because we don't just want to be creating waste that goes into landfills. Another thing is that polymer I showed you is based on, also ultimately manufactured from aziridine, which is a terrible carcinogen and not something you really want in the life cycle of your product. And then third, how to regenerate the surfaces after they've been too much covered with oil would require some kind of maybe degreasing process. And then you're just shifting the problem of oily soil removal from the laundry into this secondary application. And then if you want to use solvents to do that, then there are exposure issues. So how do you do that? So one potential way to address at least some of these concerns is to have a product as a service model, for example, where a consumer would mail the object back to the manufacturer for them to regenerate it safely, or to have something that fits into a cradle-to-cradle design philosophy where it can either be infinitely recycled or it can be composted or something like that, so that there is no garbage problem associated with it. So this is really far out, but it's bottom right corner, really far out, but it's relevant to the cleaning process, and it's our proposal based on existing science.
All right, so we just want to conclude by recapping. We think we've identified 5 pretty good strategies for our partners to pursue depending on their preferences. If the interest is more on the chemical substitution level, we think we could focus on biosurfactants or bio-based solvents. The formulation and process levels would involve the nades, enzymes, and the oil adhesive surfaces. And our goal here was to create an opportunity map for our partners that, with different strategies that would fall under implementing now versus in the near future versus further along down the road, more research would be needed. And we think the bio-based solvents could potentially be implemented now. The biosurfactants and enzymes would be strategies that could potentially be implemented in the near future if all went well. And the nades and oil adhesive surface in particular would definitely need more thinking and research. So I want to thank you for listening today, and I want to particularly thank our partners, Method, 7th Generation, Amyris, and BioAmber. Kai Johnson's here from Method. We've been... Oh,
From 7.
We've been really impressed with their commitment to not only having high-performing products, we all tried different products of theirs, but also their commitment to really thinking progressively about what ingredients to include in their products that are better for the environment and are better for human health. So thank you for your guidance throughout the process, and we hope that some of these strategies will be useful to you going forward. Thank you.
Thank you very much to the team, and we have a chance for questions for them. Marty will kick it off.
I'll kick it off. I'm curious, you showed that some of the biosurfactants, or one potential next step for the biosurfactants, would be to work with Amyris to see if they could express it in a host organism in yeast. Is there any relationship between the pathways that they currently work on and the pathways that are needed to express surfactant?
The short answer is we're not completely sure. They have the yeast model, which is generally a model that we use for solubilities, but that particular pathway, the malevolentase, is that what it's called?
Yeah.
That particular one, we're not sure if it's optimized for solubility production. So that's like the area, the specific area I think that would need more research right now.
It'd be great to ask them for evaluation.
Yeah.
Yeah, it's a good question.
Yeah.
So I know this is a chemistry class, not a biology class, but did you consider bacteria? Because different kinds of bacteria actually eat soil and things in soil, and so.
Well, actually, I guess the way that a lot of bacteria eat soil is through these secretions of surfactants. So that's kind of the bacterial angle we took. Does anyone else want to respond to that?
We didn't, to your point, we didn't actually consider adding colonies of bacteria to detergent. We used it as bio-inspiration.
And I have a question. You just mentioned that this is a chemistry class, and I remember from the last round that there were very multidisciplinary teams. Can you tell us a little bit about your backgrounds?
Sure. Actually, we have one chemist on our team, and then the three other people on the team are actually in the School of Public Health, the Environmental Health Sciences Division. So we come more from a public health focus.
But correction, I'm also a social scientist.
That's true. I like to think of it as a chemist. You can answer all your chemistry questions.
Every one of us going through
And you know, we do have various backgrounds before being in this program, but Kira did chemistry for her undergrad, and so you know we have diverse backgrounds, but that's sort of where we are right now.
Thanks.
So those were really awesome strategies, but it seems like there might be kind of a challenge in terms of compatibility with a few select ones. You talked about like using lipases or enzymes esterases, and then another class of solvents you discussed were esters. And so I was wondering to what extent like maybe we could combine these things in one formula and taking the biomimetic approach there, a lot of organisms solve that problem by compartmentalization and keeping these different ingredients separate somehow in the membrane, something like that. So I was wondering if that came up as.
Good question.
That's a great point.
I think that's a good question. Maybe we sort of stepped around it a little bit, because what we did is thought about formulation level approaches for different groups of them, like the solvents and then the surfactants, but we didn't think about combining them across those groups necessarily because of the issues you raised.
Yeah, some of the approaches are more mutually compatible than others, that's for sure. Yeah.
Yeah, I just want to say, you guys did an awesome job. When we first met, the first day, we were talking about what ideally the project would be. It's kind of like I was describing the impossible is like, let's go broad and let's go deep. And I think you did both really, really well. I'm particularly amazed at some of the out-of-the-box thinking there, so great job. I have a bunch of questions, but I'll try to stick to just kind of a general one. As you're doing this research, what were maybe the one key question that really kind of still bothers you that you don't understand, that is puzzling to you?
I think for me it was understanding there are so many ingredients in the formulation, and thinking about maybe we don't even know how the current ingredients all interact together, so thinking about exactly how these new strategies might work is difficult without actually testing it. And there's only so much, hypothesizing and thinking based on what you've read and you know how they might interact, but without actually testing it, it's hard to know. So that was difficult to think about.
Yeah, so if you had a week of time in the lab with all the materials, what would be that experiment you would do?
Probably solicit guidance from our Professor Marty. and then we would be able to give you some concrete information.
One of the areas that puzzles me is that that water interaction on the first eutectic solvent that you talked about, and some of the possibilities there. Did you find anything around the speed of how fast that happens? Is that very quick?
How fast they would maybe break apart with increasing water content? I didn't see anything on the speed. I think it was, there was no addition of water. The nades were kind of made, and then with that amount of water content, and then applied to the solutes of interest. So I don't think it was like the nades were added and then water added. It was like a separate kind of solution that they
But that was definitely a creative idea of bundling the process and talking about the pre-treat as an initial step to get around that. So that's very clever.
I still have a lot of questions about how the nades actually works. Yeah, I mean the kinds of testing done in the literature are asking different questions from the kind of questions we want to ask. So that's where the gap is, and so that's definitely something that we would need to look at in the lab.
I'll save the rest for the questions.
So I also want to make a comment, make a question. Well, first, great job, everybody. Kudos to the Berkeley students who are well known to be creative. So you certainly have thought out of the box and come up with more than one. And to echo what Kai is talking, it's one of the areas that is certainly challenging. And I think that the fact that you were able to group people from totally different disciplines, 3 from more the health science, with one chemist, and you guys worked well, probably better than we can do from a multidisciplinary team. I have 2 additional more on the comment. My understanding on this project is you're trying to look at alternate, more innovative way of cold water cleaning, right? And so my challenge in terms of thinking about that is that I think cleaning in cold water is difficult in the first place with conventional cleaning. The conventional surfactants that being used with the conventional brands like Liquid Tide, Whisk, and so forth. Right? So you have one challenge is in terms of how to improve cold water efficacy. Then you have the other challenge, which is how do you do it with the more bio-based chemicals. Have you separated the two? Have you been able to say that, forget about the bio-based for a while, how do I improve the cold water cleaning in the first place? And then take it a step further to say that what if we wanted this to be more bioeconomy with the bio-based surfactant, bio-based solvents, and so forth?
That's a really good question.
Yeah.
I would say for the biosurfactants that it was never really separated because it seemed like, because there's such a wide diversity of surfactants in nature that operate at like different temperatures, like they can be optimal at different temperatures or different environmental conditions, that it already appeared to be potentially, they could potentially be more specific for having, or for the low temperature wash cycle. And then they have this added benefit of being bio-based. Does anyone else want to add to that?
Yeah, I think that bio-inspired design was one of our core methodologies. So that was what we wanted to use to lead us to the cold water solutions. We didn't think separately first about the chemical engineering or cold water aspects, and then look for ways to make it green or bio-inspired, just because we wanted to try using that as the research method.
You want to do it all together, one step.
Yeah, well, we had encouragement and good examples from our professors. So I've never done that before, so I don't know about you.
I would just say that I mean, they did a beautiful job of executing the approach that we tried to do, which is instead of bringing in the environmental performance at the end, we put it up front as part of the challenge. That the whole, to make environmental performance and human health, safety in the key design criteria, not to do technical performance first and then say, oh, by the way, what is this going to do to the environment or to human health? And they really took that up and then ran with it beautifully.
Yeah, good job.
Thank you.
Yeah, I had two questions. One, I think was on a lipase. You said had it took longer to work, and I'm wondering, was that a difference that had to do with that could actually be beneficial in a wash cycle? I'm wondering what the time scale is there, because in some ways you could think about if something is actually working throughout the entire wash cycle instead of the first 5 minutes of a 40-minute cycle, that's a good thing. But I don't know, are you talking about the difference between 10 minutes and an hour, or 10 minutes and 3 hours, or, I was just kind of curious. That was my first question. And then the second one was, I realized you looked a lot at the hazard profile. Did you look at all deeply in terms of the life cycle footprint of some of these, in terms of the production of the enzymes or production of some of these biochemicals? I realize a lot of them seem to be experimental or in pilot stage, so we don't know what the full scale up would be. And the thing is, the chemical side, the fossil chemical side is pretty well characterized, but I'm just curious if you looked at that life cycle at all.
So to answer your first question, the research shows that lipase continues to work for multiple washing cycles. Some manufacturers cite this as a benefit because but really in terms of producing an odor, it really isn't necessarily beneficial. That's where potentially ingredient formulation could be maybe used to inhibit its activity at a certain point. It can continue to work if it has feedstock available.
Okay.
And then to answer your second question, we were limited by time to really get into some of the deep life cycle assessment, but it is definitely something that we've considered, and we will be including a lot of that information in our final report. Does anybody else want to add to that?
I would just add one thing about it, which is that it's a very relevant question of comparing, especially because what they're trying to do is develop something that works at lower temperature for energy saving and purposes. And that the tools that already exist for life cycle assessment are well developed. And what we're encouraging them to do is start exploring hazard assessment, for which there aren't great tools. And it takes a lot more legwork and detective work to bring that kind of analysis in. So part of it is just the scope of what they can accomplish in a semester, and we're asking them to do this really hard piece of hazard assessment.
I got, and I think it's curious to me, like some of the ones that are the bioengineering, it's not even clear that the current life cycle tools are well, because nobody has put a lot of information about their energy throughput and how much do they actually need compared to a conventional, fossil fuel formulation process. So those may actually be a big black hole as well, because nobody's put in the data.
That's actually a real key part of when we do the formulations. We put a lot of energy into thinking about that. The light-based one's a really interesting one. It actually has very strong effects in a short time frame, but its front combustion, the odor issue is just horrendous. It's really, really bad.
So I had a question for you about water chemistry, which I know is an important part of sort of detergent chemistry, but not something that's really talked about a lot. And depending on how hard the water is, these cations can really disrupt the surfactant effect. And I know at the beginning you called out EDTA, but then you didn't really mention an alternative chelator. I guess you had citric acid in there, which is a strong natural chelator, but an order of magnitude less. So I was wondering if you thought any about how this would vary regionally, and if you could come up with a greener chelator and things like that.
Yeah, as far as we know, we didn't really look at chelating calcium and magnesium as one of the core parts of this challenge, just because we sort of thought of it as a problem that's already being addressed by other ingredients in the formulations. And the companies that we worked with, 7th Generation and Method, have already found alternatives to EDTA. And there are a variety of different regionally diverse alternatives like that. That multilamellar vesicle that Julia showed, the reason that's important is to suspend zeolites, which are not soluble, but they work to absorb those cations. So there are a variety of non-EDTA ways of doing that. But you're totally right. Another research direction that I guess we should add, especially for the biosurfactants, is to look at the salinity and water hardness effect on how well those perform. Yeah. You had a question?
I think, first of all, awesome job. Very nice and thorough thinking. Actually, raised a couple of ideas, obviously. One comment I would like to make, and I don't expect you to have an answer to that, it's just that, a way of thinking about these problems is that, one of the challenges in the laundry is that the ratios of them, the concentrations of the substances that land in the washer is just very, very low. So, and then you pointed out that these new solvents, you would rather not use it in a laundry where it gets diluted and minimizes the effect. The same could be the eutectic material problem. But if you approach the thinking from if you imagine that you would design a delivery system rather than just get all of your materials dissolved, like we are very well aware of the fact that drug delivery methods can make or break the efficacy of a drug. So thinking on the same line, if you were to think about how these solvents would behave in a surfactant system, which you pointed out forms micelles or other aggregates, that might keep your solvents intact, and it might actually deposit them on the surface of the fabric where we want them and not get dissolved in the water. So that might change the whole picture. So that's one observation or way of thinking about these systems. The second thought is that the same way this association or or delivery system might help us out in the laundry, that might also change the outcome of the biodegradation and the safety. So I was wondering if did you find anything in your literature search about that, how that plays out, or whether the safety, environmental safety, is different depending on the other components or the other environmental compounds that are present. It's just.
That is a great point. Thank you for bringing that up. We hadn't really taken the next step to think about new ways of delivering the NAIDS materials in the context of a laundry wash. The only thing is the pre-treatment strategy. That was our one attempt to do that. But that's a very valid point, and yeah, it would definitely have it would necessitate another level of hazard analysis. Kira, do you think?
No, I think that's a really. We have another strategy now.
Yeah.
Thank you. But yeah, we haven't thought about the next level of interactions of those, how the ingredients might interact in the environment. So another thing to consider, definitely.
No other questions, we'll move on to the next group and congratulate this group on their work.